So, hello everyone. We are really glad to see you here online, even though it is really great weather outside. Today we are going to talk about high content imaging for drug discovery. Our lecturer, Viktoriia Kosach, is a great specialist in this topic with more than 5 years experience in various CROs in Ukraine and also in Poland. Feel free to ask your questions in the end of the lecture. We are going to have discussion and Q&A session. It will be both in Ukrainian and also in English, so you can ask whether you are more comfortable. Also, in the end of the lecture, you're going to have after the lecture we will send you feedback form with this recording of this lecture and also with the transcription of the lecture translated to Ukrainian. So I'm going also to repeat the same in Ukrainian, and we will start soon. Дякую, що доєдналися. У нас сьогодні буде лекція англійською про High Content Imaging у розробці ліків. В кінці лекції у нас буде дискусія і питання та відповіді. Вони будуть також українською та англійською, тож ставте свої питання в чаті або включайте камери чи мікрофони і задавайте свої питання Вікторії. Також після лекції ми надішлемо вам транскрипт цієї лекції, перекладений на українську, і також буде доступний запис, і все це ви зможете отримати, заповнивши фідбек-форму. So, Victoria, please, the floor is yours.
Valeria, thank you for your kind introduction. And hi everybody, I am really happy to see you today on our seminar. It will consist of two sections. At first, we will take a look at the general information about high content imaging, its application, as well as some tips for assay development. And at the second chapter of the seminar, I will show you some real-life examples of high-content imaging assays. So let's start our lecture. I believe that most of the registered participants are already here. Okay. Valeria, could you confirm, please, that the slides are moving and changing?
Yes, everything great, and also I see your laser pointer.
Thank you. So let's start from actually a definition what is high content imaging. So it's general automated microscopy-based assay that is usually used in drug discovery and industry to simultaneously measure multiple features in single cells, usually after treatment with some perturbation agents like small molecules, siRNAs, or CRISPR libraries. If you will go further to the literature, you will also find some other terms or phrases, and these are high content screening. This one refers to the large-scale project when you need to analyze more than 100,000 compounds, even up to 500,000 compounds. The second one is high content analysis. Usually it's used in the same meaning as high content imaging, but from time to time, it could refer only the analysis part, analysis workflow of the data. And the last one is image cytometry. It's really rare, but it's just a synonymous of high content imaging. Let's try and compare the research microscope and the automated high content imager. So here on the left you can see the general classic size laser scanning confocal microscope. You could see that it has a lot of possibilities to manual adjustments. However, there are a lot of advantages of research microscope. One of them is possibility to use high numerical, high resolution immersion objective lenses. For example, oil objective lens with resolution of 100. And usually these objective lenses are limited or not available for automated imager. Because of that, research microscope could give you a very nice resolution of the images. And actually, there are a lot of better possibilities to separate the spectral overlap. So currently, research microscopes have very nice software to work with fluorescent dyes that have a very tightly packed or even overlapping spectral excitation emission. So after the experiment, you could better separate them. However, for regular research microscope, to publish a data, you will need to image around 300 cells per one condition. And actually, it takes time. It takes longer time in comparison to automated microscopes. So actually, such microscopes, as you can see on the picture, is just usually a black or white box where you couldn't really manually change something there. And they were developed to be highly unbiased or highly non-biased, because you could not pick a cell that you like. The microscope will just go throughout the whole field of view and take the whole cells in unbiased manner. Except this, the automated imagers were developed to be much faster than the research microscopes. For example, you could read the 384 well plate in 30 minutes or in 1 hour, depending on your setup. But actually, believe me, it's really fast image acquisition. And also, I already told you that these microscopes usually operate with multi-well plates to increase the throughput, to increase the amount of compounds that you could test in one run. And for industry, you should take for one condition at least 1,000 cells, maybe 500, but at least 1,000 cells per condition, usually even more. So such microscopes give you very high statistical power. However, automated high-content imager could also be used for small-scale research projects because they have a number of adapters to use different sample types. So it could be not only multi-well plates, it also could be classical tissue samples, classical cells grown on cover slips. It could be even Petri dishes for live imaging. However, these microscopes are a bit limited in the objective lenses that you could use, because I did not hear about such imagers with oil immersion because it's very complicated for automation. So that's why such microscopes usually have water immersion objective lenses. They are also not such quick. These objective lenses produce a bit higher times of acquisition. So that's why engineers made an objective lenses that are known as high aperture, high numerical aperture, but dry air objectives. And usually these ones are the first choice for high content imaging assays. So let's quickly look how these imagers are such quicker and faster than regular research microscopes. So on the left side you can see a picture of wide field fluorescence microscope. Both research and automatic ones could operate in such a type of microscopy. So I think you know that usually a light source is a lamp that excites the whole field of view, and the emission the fluorescent emissions is also collected from the whole field of view, different focal planes. So in such a way you receive a very intensive, very nice intensive signal, but it has a lot of out of focus light from different focal planes. In confocal microscopy, to avoid this background light, there is an aperture filter that has a pinhole in it that focuses the light in one very small point in one focal plane, and actually it block out the fluorescent emission from the background or non-focal plane. So in that case you receive a very nice image, like very strict, very detailed. And confocal microscopes, laser scanning confocal microscopes, just scan your sample using this pinhole point by point in a focal plane. In automated imagers, to make it a bit faster, they did a pinhole, not like a point, but rather like a line. So it's scanning your sample line by line in one focal plane. So such the image acquisition is faster in comparison to research microscopes. There is a bit other approach. I think maybe you heard that there are confocal microscopes that are called spinning disk microscopes. These microscopes have the same aperture with pinhole, but actually a lot of pinholes on the disk that is moving and collecting this light from the higher area much faster. Actually, these microscopes are very nice for live cell imaging because they are fast and less toxic. And there are research microscopes like that, and there are automated microscopes from Perkin-Elmer, for example, with the same technique. However, engineers went even further, and on the market there are actually spinning disk automated microscopes with two spinning disks that are working simultaneously, enhancing the light that they could collect and even more faster than the other ones. Such microscopes are produced by Yokogawa, and as far as I know, one of AstraZeneca facilities operate with such double spinning disc microscopes. So as we can see, such very fast and high throughput microscopes are really nice to use in large-scale drug discovery process. Let's look at what stages of drug discovery high-content imaging could be used. To be honest, in almost all of the stages. So the first stage of drug discovery and development is target identification and validation. So you want to know if or which protein, or if the protein of your interest is really involved in developing of cancer or other disease type that you are working with. To assist it, to confirm it, you would usually do some knockdown or knockout assays using siRNA or CRISPR library. However, in some cases, you will need to overexpress your target to see the difference in cell behavior. So for all these assays, high-content imaging could be used. It's less scale, so small-scale experiments, more research-grade experiments at this stage of drug discovery. However, when you already know your target, you would like to find the compounds or other chemical agents that could influence, inhibit, or activate your target to prevent disease development. For this, there is a stage that is called high-throughput screening. It's a large-scale testing of compounds starting from 50,000 and to 1 million in AstraZeneca also, for example. At this stage, you will use different functional assay, different microscopy assay. You could just imagine or find in literature for your particular target. For example, it could be translocation assay. When your protein is active in nuclei of the cells, but inactive in cytoplasm. So you could measure this flux or movement of your protein under the treatment with compounds. You can do regular intensity measurements to see the expression level of your target. You could count the number of lysosomes, autophagosomes, nucleoli, so any organelle you are interested in. And you could make some colocalization testing of your target with a marker, for example, with mitochondria marker, or the other compartments where your target usually could be found. The one thing about the high-throughput screening with high-content imaging is that the features that you are analyzing are very, very reduced to about 2 to 4 main features only because of the large scale, because of the time saving of this phase of drug discovery. After the high-throughput screening, you will receive some amount of compounds that inhibit, for example, inhibit your target. These compounds are called hit compounds. So you take them to the next step, and it's called hit-to-lead development or lead optimization. So from these hit compounds, you will choose one or several that you will work further to the market if you will be successful. So during this stage, your chemical team is modifying the structure of compounds to make them more soluble, more cell permeable, or just more active. So you are playing with structure of the compound. And to test these new analogs with new structure, you also will use usually the same assay as in high throughput screening. However, you could also imply some phenotypic profiling. Phenotypic profiling is when you are just looking to the morphology, different morphology features of your cells, and comparing the morphology of the cells treated with your hit compound and to morphology of cells treated with new analog compounds. After that, when you choose already your lead compounds that you will work with, you are going to lead preclinical tests. Usually preclinical tests involve animal experiments, but before this, you would like to decrease the amount of animals that you will be using for experiments, or maybe even you will need to do some in vitro tests that will help you even to avoid the animal tests at all at this stage. So such in vitro assays with high content imaging could be multiplex cytotoxicity. So when you could compare the cytotoxicity of your prospective lead compound in your disease model, for example, cancer cell lines, and in normal cell lines, for example, in human dermal fibroblasts. So if you will see that some compounds give you the same amount of cytotoxicity in both disease model and normal cells, so you will not use it because it's not specific to your disease model. But if your compound are very specific and induce apoptosis or necrosis or other types of toxicity in cancer cell lines, in your disease that you are studying, and are non-toxic in normal cells, that it's very, very great for your compound development and drug development process. The next assay is genotoxicity assay, which shows you if your compound influence, for example, the cell mitosis or the cell proliferation, and could have any genotoxicity in vitro. This assay is usually called micronuclei test. The other example are hepatotoxicity assays, like steatosis and phospholipidosis in HepG2 or other cell lines. These are very, very important because I think half of the drugs, or a lot of prospective drugs, do not appear at the market because of their hepatotoxicity or the hepatotoxicity of their metabolites. Maybe you have read some news, some recent news, that there are unfortunately a lot of hepatotoxicity cases in patients already that are used or have used biological additives like curcumin or even some high doses of vitamins even now, because unfortunately biological additives are not very nicely tested for this. So hepatotoxicity assay is very important, and we also need to be careful with biological additives that we are using right now. And the last but not least example of the high content imaging assay in preclinical tests are assays in 3D 3D, like cancer spheroids or organoids, because 3D models are much closer to what is happening in organisms in comparison with regular 2D monolayer cultures. And actually, the next stage is already clinical trials in patients, and here the use of high content is very limited. However, still it could be used for example, blood samples or tissue samples analysis in patients. A bit separately, in parallel to high throughput screening, we can use such called phenotypic profiling or cell painting assay. This assay is usually much challenging in data analysis because this assay collects from 1 to 3,000 features from one cell, each cell in the assay. So after that, you can use machine learning or AI to find different morphological phenotypes inside your experiment. This assay could be very helpful when you have a known compound tested, and you just have to find the new analogs of this compound that will give you the same phenotype. Also, this assay is very interesting and helpful if you do not know the mechanism of action of your compound. So using this assay, you will find the features that are influenced by your compound, and in such a way you will find the cellular compartments that are influenced by your new drug, and in such a way drive some conclusions about the mechanism of action. Okay, so we looked at the applications of the high content imaging. What about assay development for this automated microscopy? Assay development will be more or less the same for all stages of drug discovery and high content imaging, with a small minor differences. So let's take a look to some important considerations before you will start with your assay. First, you know your target. So you will find a marker with which will help you to visualize your target. And also, you should know, is your target is expressed, in which cell lines it is expressed, or should you transfect the cells with your target, or is it not a single target or more a phenotypic response? After that, I think maybe the most important consideration is your cell model. So will you use general immortalized cell lines, or will it be some primary cells, or maybe it will be stem cells? And for each cell model, you should investigate the precise growth condition, and as well as validate the cell seeding density, the number of the cells that you will plate in each well of your multi-well plate. About plates. So actually the next important part of assay development is choosing the right plate for your assay. It is really very important to have good quality images. And you also should know if your cell line will need to have just tissue culture treated plate, or the plate should be coated with some matrices like collagen, poly D, lysine, etc. After that, you will find the reagents and probes for your assay. We will talk about it further. And also, quite important thing to know is the right reference compound. Reference compound is your positive control. So actually the compound that is already validated and known to induce the need influence on your target. So you know that this compound is inhibiting your target or activating, depending on the assay. It should be already published better, or just be a drug, a recent drug that is already on the market. So it's also quite important thing. And at the end, when you know all of this and you are starting performing first experiments, you will play with image acquisition and analysis to receive the best quality control measurements which are possible. So during the assay development, it is important to have quality control during the whole assay. In such a way, you will receive your final conditions with which you could proceed to higher scale, high throughput screening. And let's go briefly through some important conditions during assay development. And firstly, as I have told, it's your cells. The health of the cells will influence your assay like a lot. For high content imaging, the most classical cell lines that are widely used for theoretical studies more, let's say so, are U2OS, it's human osteosarcoma, and A549, human lung adenocarcinoma cells. They are used in such image-based assay because these cells are very large, they are flat, and they have a lot of the cytoplasm area in which you could define your organelles precisely. However, these cell lines are not good for any disease models that you are interested in if it's not oncology. So you will consider the other ones like primary cultures or IPSC. But you should remember that some cell lines, especially normal cell lines, for example, like human dermal fibroblasts or human embryonic kidney cell line, they have very small amount of cytoplasm, and it will be really challenging to detect any changes in cellular compartments if you will need. So it's better to look to the other cells, or maybe to try to use any coatings like collagen, then these cells could spread a bit their cytoplasm, so you will be able to detect your features. Important considerations about cell seeding density, so the number of cells that you put in your plate. So usually it should be 80 to 90% of confluency at the time of imaging. However, you should also check at the high confluence level that your cells are not like squeezed or are not very densely packed to each other. Because, for example, with overconfluent or 100% confluent cells, the shape is starting to change. And also in such a situation, your image analysis would not be able to detect as separate cells. So it is very important that your image analysis could separate the cells. And you could also assist the distribution of the cells in one well. So it should be even distribution because some cell lines, for example, could stack and cluster. For example, HepG2 and MCX7 cells, they could cluster, and then image analysis are also not working very well. In such cases, you could use cell strainers or cell filters. So you filter such suspensions through it, and you receive more or less single cell suspension that is better for high content imaging. Next, about the cells. So, as I have told you, the cell health is very, very important. And some parameters, growth parameters, could influence it. For example, change of the serum, of lots of serum that you are using from the vendor could influence cell growth and answer to your treatment. So please, if you consider a large-scale experiment, you should be sure that you secure the enough reagents, enough serum to the whole experiment. So you are not changing your reagents during your experiment. And I think you all know it, but still I need to say that your cells should be healthy. They should not be contaminated, especially with mycoplasma, because mycoplasma change growth rates, metabolism of the cells. It could even influence the response of your cells to the treatment, to your prospective drugs that you are testing. And moreover, if you can see here, so mycoplasma on the images during high content imaging are very visible. It is visible, and this mycoplasma staining will actually influence image analysis. So there will be artifact in your image analysis. Okay, and maybe I think that one more thing that I will tell you is the importance of passage number of your cells. It is required to have no more than 20 passages of your cells, especially if it's transfected or stably transfected cells, because after 20 passages, your cells could change in nature, in genetic material, so it could influence your assay. And the second very, very important feature in assay development for high content imaging is plate type. One of the best for image properties are glass quartz bottom plates. They are flat, the most flattest, and they are very nice for high numerical, high resolution objective lenses. However, they are very expensive and they need coating, and they are also quite fragile. So actually, from my experience, almost all high content imaging scientists are using cyclic olefin bottom plates. It's a polymer that also may help to make very flat bottom of the plate, and it works quite good without any coating, and also could be used with different objective lenses, which is also very important. However, they are quite expensive. And polystyrene plates are the classical plates that are used in other assays. They are not very nice for imaging. However, they could be used with small resolution, so with the smaller objectives like 2x, 4x, 10x even. So they could work with them. So actually here you can see the flatness. Here you can see the flatness of the bottom of different plates. So this is the regular polystyrene plates. You can see that there are very high variance in bottom thickness. While in glass bottom plates and cyclic olefin bottom plates, the variability of bottom thickness is much less. Actually, I like a lot the best results I got with cyclic olefin plates from Revity. It's the old Perkin-Elmer distributor with plates that are called phenoplate. Except the cyclic olefin bottom, this plate also has a very low high of these bottom borders. Let's call them like that, because in such a way, all the objective lenses could reach all wells in the plate. Because in regular plates, like polystyrene plates, these borders are much higher, and this bottom is also higher in the plate. So objectives like 40x, 60x, 100x could not reach, because of their physical parameters, they could not reach these border edge wells. And a bit more about plate type and some effects that could influence your assay. It's the evaporation from the plate or such a called edge effects. So you can see these columns is a representation of separate wells in the plate. So this is a regular plate with regular cover lid. And for 24 hours, this is evaporation level from these wells. It will influence your assay very much. And here on the right hand, it's the evaporation level from the wells in this Revity phenoplate. It has a bit different plate lid that lowers this evaporation. Evaporation level and edge effect on your assay will depend also on your CO2 incubator, because all incubators have some gradients in temperature that could provide you with such edge effects. So actually for your assay, if it's not a very large high content screening, but if it's a small scale experiment, it is very recommended that you do not use these edge wells for your experiment. Instead, just put there the PBS or gross media without cells to lower the influence of evaporation on your data. Which is recommended to firstly use some commercial kit that is validated, that is provided with exact concentration of fluorescent dyes. So in such a way you could quickly adjust your protocol steps, and after that, if you have a large scale and you would like to pay less, so after that you could just buy these fluorescent probes from the kits separately to make something like homebrew homebrew reagents, and validated compared to the commercial. You know, your fluorescent dyes or fluorescently labeled antibodies expression emission spectra. I highly recommend the fluorescence spectra viewer from Thermo Fisher. It is very nice to play with this website. You could add your fluorescent props on here, and you will see right away if you have any potential problems with overlapping of the spectrum. So actually for high content assay where you will use several fluorescent dyes, it is recommended to choose the dyes with the spectrums that overlap the less. For example, like here, the dye with the green emission and with far red emission. So overlapping is much less when you in comparison with green and red or orange emission, where the overlapping of the spectrum are high. So fluorescence spectra viewer, very useful to design your fluorescent probes for the assay. When you have all of this established, you are going to your confocal microscope for image acquisition. I think you could know that actually image acquisition is just a digital value from the light emitted and detected by the detector or camera on the microscope in gray values, and this digital parameters actually are quantifiable. So each pixel have its own number of gray value. And you should know the so-called dynamic range of your microscope. So for example, human eye dynamic range, so we can distinguish around 500 gray levels only. The old camera could distinguish only 256 different gray values, but the microscopes, automated microscopes, they usually have around 16 or higher bit camera, so they can display and detect more than 60,000 gray values. So when you're capturing your image, you want that your signal, fluorescent signal, will not give you higher signal rate than this, because then your camera will not be able to detect the all signal from your sample. So this is quite important when you acquire your images. So when you already also already can go for imaging, you will be interested in what controls you should use for your high content image analysis assay. So first of all, it's negative control. It's just the cells without any treatment that are growing in growth media. This control is very important at the first stages. However, when you are going further for large-scale screening, you will need only vehicle control. It's your cells treated with solvent. So each compound or each chemical perturbator that you are studying, it should be solved in something, and different compounds are solved in different solutions. The most common one is DMSO, but it could be ethanol, it could be water, it could be other things. So you need to have a control for just a solvent, just a % of solvent in your media, to see if it does not influence on cell health. And the other control for high content screening is positive control. It's the cells treated with a reference compound, which is clearly you know that it will influence your target in a way that you are looking for. When you have the experiment with all these controls, you could proceed to quality control check of your assay. There are usually three parameters that are very important for quality control. The first one is Z factor. It actually shows you the robust, how your assay is robust, and how it is reproducible. So actually it shows you the difference between max and mean signals and their standard deviation, so variability of the assay. Here is the formula for the Z factor. And you can see here is our max minus mean signal, and here is our standard deviations. Usually in high content imaging, that factor should be higher than 0.5. So that factor should be 1 or 0.5, not less. Then this assay is reproducible and robust. So if you will look at the formula, you will know that if something is wrong and your Z factor is lower than 0.5, so the assay do not perform well. So what you can do, you could try to increase your max minus mean, so use signal window of the assay, or you could try to lower the variability of the assay. I will talk about it in a minute. So the second parameter is signal, so-called signal to the ground. It's your signal window as the assay, but actually it's the same as max to mean signal from the signal controls. For example, if you are testing inhibitors of your target expression, for example, then your max signal, it will be vehicle. So the normal cells treated just with the solvent, they will have maximal signal of your target. And the minimal signal will be signal with reference compounds that inhibit the fluorescence and expression of your target. The higher, the better. The threshold for this is 2. So signal to the ground for good assay should be always higher than 2. And the third parameter is a reproducible SC50 or IC50 of your reference compound. So from day to day, your cells should your reference compound. So if you would like to improve the quality control of your assay and to reduce the variability, you could try to lower the standard deviation of your assay. Usually, to do it, you will use automation of your assay. You will not do it only by your hands. So you will include some robotic machines, robotic instruments. For example, there are several options. For example, on the right here is the automated high content imaging microscope from Elmer. And except the microscope, the platform has a robotic arm and plate holders. So if your assay is fixed with formalin, yes, not live, so the cells do not need CO2. So you just put your plates with cells to this stack, and a robotic arm could automatically transfer it to the microscope. In such a way, we were able to do imaging almost 24 hours each day. The other automatization possibilities are using liquid handler, acoustic liquid handler called LabSite Eco for compound distribution. So you distribute your compounds in nanoliters to the plates without diluting them manually. After that, you could dispense your buffer in the plate with compounds using a multi-mode dispenser. You could also plate your cells with such a dispenser. It will greatly reduce the variability of cell distribution in the plate. It could be a plate washer. Plate washer is especially for cells very gentle. It add and take out from the plate different buffers. Or if you are using antibodies and you need a lot of washes between the antibodies, so plate washer is also a nice option. And it could be a robot called plate replicator when you have a plate with compounds and a plate with cells. So you just can replicate your compounds on top of your cells and do not do it manually to avoid any mistakes. Okay, so we already have our assay, and one of the last steps are image analysis. So all automated microscopes, they have inbuilt software that came from vendor that do a first stage of image analysis. It consists of segmentation of the objects that you are analyzing. So usually, firstly, you will detect the nuclei in the cells, then you could detect the whole cell and organelles that you are interested in. Actually, this software would calculate for you anything you want, like intensity, texture, regions, locations, shape of the object that you are interested in, and it will give you a Excel or CSV file as a readout, which you can further analyze using R or Python, or you could move to the other softwares. Almost all automated microscopes today also have inbuilt supervised machine learning. That is very convenient if you have different subpopulations. If your cells do not answer to the stimuli like one, but there are different populations that answer a bit differently. In such a case, supervised machine learning worked in inbuilt software to the computer, to the microscope, worked very nicely, so very good. So if you would like to analyze your data further from Excel file or even from raw images, you could use some free software available like Cell Profiler, Fiji, IC, Napari, etc. They are quite a lot today. Cell Profiler is very nice. It also is very convenient for phenotypic profiling, because this software, together with phenotypic profiling cell painting kit, was developed by Anne Carpenter, so the same person. And there are a lot of image analysis workflows on the GitHub and on their website dedicated to phenotypic profiling. Okay, so actually it was our first chapter, first section. I already see that we are talking about an hour. I will just quickly, maybe not to hold you further, I will quickly show several real-life examples of the assays. Just a minute. An example of high throughput, high content study with 100,000 compounds testing. It's about 330 plates to be tested. The model where U2OS cell line transfected with target protein fused with GFP. So we did not need to label anything. It was already a fused fluorescent protein. The target was a transcription factor. It was active in the nuclei of the cells, but upon modification, the target was inactive and localized in the cytoplasm. So the goal was to find the new compounds that could activate the target. High content screening, high throughput screening projects, they are managed in cascade manner or waterfall manner. So it has step-by-step development. And first part was feasibility study. So we had to optimize the imaging, assess the QC, test the solvent or vehicle tolerance of the cells, etc. And then during main study, we had to test at the beginning a small amount of the library, reagent compound library, and then it was a primary screening of the whole 100,000 compounds library. After that, there was some confirmatory screen, and when we localized the hit compounds, our client reordered the compounds from fresh powder, tested them for quality of the compounds, and we ran those response study. Okay, so assay development. Firstly, we had the solvent control or vehicle control. It was DMSO, and we had our positive control. It was a reference compound that already listed in literature to influence and activate the target. Firstly, we tested two modes, so wide field fluorescent, not confocal, and confocal mode of the imaging. Both of them, as you can see, I hope, so as you can see, both modes of imaging produced that factor Z prime higher than 0.5, so it's a good assay. But you could also see that in confocal mode, the signal window, the difference between our vehicle control and our positive control, so the signal window was higher at confocal mode. So we choose this mode of imaging because during very large screening, something could go wrong, some variation could appear, so that you would like to stick to the higher window to have some possibility of this fluctuation happen and do not influence your quality control. After that, we polished our image analysis. So first, as I told, there were segmentation to nuclei and to cytoplasm. After that, we applied some filtration to our data and to our cells. We excluded from analysis cells touching edges. So the cells that touch edges of the images because they will produce not relevant, some clipped data that could influence the analysis. Second, we filtered out apoptotic or mitotic cells based on their shape, so they will be round, and intensity of DNA, because the DNA will be very intensive signal. So we also filtered them to not influence the ratio between cytoplasm and nuclei signal. And we filtered out GFP negative cells that do not harbor our target. After that, we collected the intensity from nuclei, intensity from cytoplasm of the target, and make a ratio nuclear to cyto intensity ratio. However, these cells could have a different answer to the stimuli, a bit different. So we had to know which cells are actually positive to this translocation to the nuclei. So we applied some thresholding to the cells. So we considered positive for nuclear target activation the cells that had higher ratio, nuclear to cyto ratio, in comparison to DMSO control plus 1.5 standard deviation from DMSO control. This number is usually 3, but our client, after literature search, they wanted us to have a bit less threshold in here. And the last step, actually this step is also very important. You should normalize your raw data somehow. Not somehow, there are 3 different actually possibilities to normalize the data. But usually you will transform your data into percentage. And in this case, the percent of translocation of the target, positive cells with translocation, was the number of positive cells divided to total number of cells and multiplied by 100%. In such a case, when you normalize your data to the percent, it could be percent of your control, it could be percent of inhibition of your target, could be percent of activation of your target, etc. But it's really important because from day to day, your microscope and your laser could perform a bit differently, depending even on the temperature in the room, in the laboratory. So your raw fluorescent units will a bit differ between day to day, maybe between plates. But your effect should be the same. And if you normalize to the percentage of the cells, you will discard these small differences between raw numbers day to day will be eliminated by normalization of the data. Here you can see the next step was studying the solvent influence on your assay. Solvent was DMSO in this project, and here you could see the cells without DMSO with the target in cytoplasm, and then you see the images with DMSO with different percentage in gross media. So 2% is very high. You can see that the cells change their morphology. It's usually due to cytotoxicity. 1% DMSO already has some cells that change morphology, but it also influences a bit to our here you can see the nuclear to cytoplasm ratio. So this is 2% DMSO, 1% DMSO. So they influence our assay without and should be excluded. So for further screening campaign, we could use only DMSO up to 0.5%, or better, even less, so up to 0.25%, not to have any artifacts from our solvent. Okay. After that, we started our high content imaging campaign. You could see an example of generated, this is reference compound IC50 dose response curve. It was included in each plate as a curve. And these are final quality control. So flow control, vehicle control, and reference compound activation control, and that factor was about 0.94 after image analysis optimization. And the results, the conclusions. So on here on the graphs, you can see IC50. Each dot corresponds to tested plate. So here we have around 300 plates, signal to the ground, and Z factor. So our threshold for Z factor, as you remember, is 0.5. And you can clearly see that during more than a half of the screening campaign, everything was good. But here in this, at the end of the campaign, something went wrong. Some plates did not pass the quality control, as well as at the more or less the same time, we had a break here in IC50, did not reproduce. So we had to repeat, to redone these plates. And actually together with the team, we had two thoughts what went wrong, and actually we still do not know what exactly it was from these two variants. So first, when something like that happened, and you see that IC50 change, you could think that something happened with cells, because cells started answer differently to your control compound, to your reference compound. So this is one hypothesis. And the second hypothesis is that nearly in the same time, the robotic arm that transferred the plates to the microscope had broken. So it was broken, and the plates had our high throughput went down. We did not have a possibility to process 24 plates per 24 hours without the robotic arm. And the plates were seated in the plate stacker for several days while we were trying to do something with robotic arm. So it also could be problems that do this. But as a result, we have screened 328 plates, more than 100,000 compounds. From this library, 100 heat compounds were selected with translocation higher than 30%. Hit rate is quite low in this assay, but it's because of the target. This target is hardly druggable. And 6 compounds from 100 were excluded from hit list because they had very high toxicity. So in parallel to all other measurements, we assist the toxicity by calculating the number of the cells, the number of the nuclei in the well. So this is an example of a huge campaign, microscopy campaign, where high content imaging could be used. And very quickly, so I will show you just I think, two more examples, not to overload, as always, the information, for other stages of high content of drug discovery. So as we already discussed, high content imaging could be used for target identification with siRNA or CRISPR library. So here you can see two cell lines that are sensitive for with sensitive to target. The target is member of DNA repair machinery, essential for survival of some cell types. But the other cell types that are non-sensitive, they will still proliferate. So we used siRNA in different concentration to lower the expression level of the target and to see if there will be still DNA damage in the cells. We use DNA damage marker for it, and as you can see that we in the siRNA control, so in some target sensitive cell line, so in target sensitive cell line, we see the formation of DNA damage locuses in the absence of the our target. And here, actually, after data analysis, we evaluated and received very nice dose response in a sensitive cell line on the concentration of our siRNA that showed us that the target indeed is important in this cell line. However, in the cell lines that are non-sensitive to our target, we did not see any response, any DNA damage locuses enlargement, and no dose response. So for us, it was a clear example of the target involvement in DNA damage in sensitive cells. And for this assay, we just segment the nuclei and counted the number of DNA damage locuses inside the cells. And the last example for today, it will be the high content imaging at the preclinical in vitro studies. And you can see the liver hepatotoxicity assay using HepG2 cells in 3D. So the spheroids of the cells were treated with cytotoxic reagent compounds, and you can clearly see the difference in spheroid area as well as the number of live green cells and red dead cells upon treatment with this compound. So these compounds are for sure toxic for liver spheroids, as well as data analyzed from these images could be visualized like dose response in here, showing the surface area of the spheroids during treatment with the compounds. And this received IC50 from our department, aligned very nicely with literature IC50 for these compounds. So this assay could be used and are very important for drug discovery process. So, thank you a lot. Thank you a lot that you had the possibility to join today. And if you still have some energy, maybe you would have questions. You could write them or you can ask them. And I would like also to thank you to the Bioinformatics for Ukraine for giving me this possibility to talk to you today.
Thank you, Viktoriia, for your lecture. So now, everyone who has questions, you can either write them in the chat or you can just turn on your mic and ask your questions. Дякую, Вікторія, за лекцію. Зараз всі, в кого є питання, ви можете їх або написати в чаті, або просто включити мікрофон і поставити свої питання. Вони можуть бути українською або англійською, як вам буде зручніше. Тож, чекаємо, якщо є в когось питання. Якщо немає, в мене є питання, звісно.
У мене є питання. Доброго дня. Дякую за лекцію. Було дуже цікаво і інформативно. В принципі, 90% моїх питань були задоволені. А мене таке цікаве, чи використовуєте ви якісь методики для детекції нейротоксичності чи нейротоксичності, в принципі? І які лінії клітин ви для цього використовуєте?
Дякую за запитання. Я дуже рада, що більшість питань були описані в цій лекції. Щодо нейротоксичності, коли я працювала у CRO Янамін в Україні, то, на жаль, у нас ще не було таких моделей, і наскільки мені відомо, якраз Валерія перша, хто почала їх розробляти у Янаміні, тому, можливо, вона трішки може прокоментувати, тому що, на жаль, особисто я не працювала із нейромоделями. Те, що я читала, це Neurite Outgrowth модел може бути застосована, але навіть для цитотоксичності, просто для цитотоксичності це також може бути такі барвники стандартні, як пропітіум-йодид або маркер для детекції Каспас. Я думаю, вони також можуть бути застосовані в цій моделі. Окрім того, для цитотоксичності таких складних моделей може бути використана якраз оцей cell-painting або morphological profiling, те, що morphological profiling. Під час цього клітини, які будуть максимально округлені, вони можуть вважатися мертвими, і їх можна цей фенотип відрізнити, на мою думку. Але, можливо, Валерія щось теж прокоментує, бо я знаю, що вона цим займається більше.
Насправді мені немає навіть чого особливо додати, бо так я почала лише з первинними культурами. Я знаю, що Романа цікавить, ми знайомі, працюємо разом, Романа цікавить лінії, які можна диференціювати в нейрональні культури, але поки що я до цього не доходила. А стосовно того, які моделі планується використовувати, то це так і є, це cell-painting, можливо, і каспазні маркери, які також на мікроскопі подібному хотілося б реалізувати. Так, я зараз поки що не бачу питань, тому я скористаюсь можливістю, задам своє.
У мене ще є питання.
Добре, Роман, якщо ти не будеш проти, я вже завершу своє. І
Вибач, що Валерія. Ну, давайте ви перші, да.
Добре. У мене питання стосовно того, чи траплялося вам використання саме первинних клітинних ліній для high-content іміджингу, І в мене це питання скоріше в контексті того, що первинні культури, якщо вони саме на нульовому пасажі, вони мають досить багато дебрису, який не завжди вдається вимити до моменту того, як уже з клітинами можна проводити дослідження і свій експеримент. Чи ви бачили фотографії, на яких це було помітно, чи таких фотографій не траплялося і, скоріше за все-таки це проблема саме мого есею, чи є якісь взагалі коментарі на цю тему?
Дякую за запитання. З первинними культурами трішки працювала моя наукова керівниця з інституту. Це на той момент були культури фібробластів, культури кератиноцитів шкіри. І дійсно на першому етапі після виділення там були ще не монокультури, по-перше, так? Тобто там могли бути клітини різних типів. Тому вона використовувала маркери для розрізнення клітин, тобто якісь поверхневі маркери, щоб бачити, де ті клітини, які її цікавлять. І все ж таки вона починала трошки пізніше, тобто не на G0. Вона давала все ж таки хоч трішки їм пасажуватися, можливо, диференціювання, не знаю, але тобто вона все ж таки їх трохи вела до моменту іміджингу, щоб максимально відсіяти ось цей дебріс дійсно. Якщо дебріс виникає не тільки через різні типи клітин, але саме через шматочки, якісь ушкоджені клітини, якісь мертві клітини, то їх можна спробувати на етапі імідж-аналізу відфільтрувати за формою або розміром. Тобто зазвичай клітини, які вас цікавлять, вони можуть бути прям більші, тоді як дебріс, якісь апоптичні або шматочки чогось, вони будуть маленькі, дрібні, і дійсно їх можна спробувати відфільтрувати.
Дякую. Романе?
У мене трішки технічні питання. От все-таки, зважаючи на ваш досвід, що краще, конфокально використовувати чи Whitefield, і мене щось цікавить, чи робили ви якийсь high-throughput, high-content imaging прижиттєво? Клітин?
Ні, на жаль, прижиттєво я не працювала взагалі, тобто я тут не експерт щодо high-throughput, не підкажу вам. Вибачте, будь ласка, чи могли б ви повторити перше питання? Перепрошую. Те, що було перед.
На рахунок конфокального і Whitefield.
Так, добре, дякую.
Конфокальний і Whitefield. От ви вважаєте це, чи взагалі потрібно використовувати, наприклад, конфокал?
З мого досвіду якраз із цього есея по транслокації з цитоплазми в ядро, то з мого досвіду ми завжди пробували паралельно Whitefield і Confocal і порівнювали Quality Control Parameters. Тобто ми пробували завжди. То якраз для Translocation Assay або для підрахунку якихось лізосом, якихось органел, то нам підходив більше конфокальний режим все ж таки. Що стосується cell-painting або морфологічного профайлінгу, то ми також трошки, на жаль, я не була залучена до дейта-аналізу, але ми оптимізували трошки протокол для cell-painting. Ми пробували конфокальний режим і Whitefield Fluorescence, і ми зупинилися, ви знаєте, на Whitefield Fluorescence, бо його було достатньо для визначення різних фенотипів. Тобто, щоб Machine Learning або AI визначили окремо ці фенотипи, флуоресцентного Whitefield нам було на той момент достатньо, плюс сама М. Карпентер з своєю командою, в них вже трошки є статей по селф-пейнтингу, то вони також використовують Whitefield. Я не бачила, щоб вони робили конфокал. Я думаю, це пов'язано ще з тим, що все ж таки Whitefield трошки буде швидший, ніж конфокальний мод, бо там дуже багато цих маркерів, так, там ще 6, мені здається, fluorescent dyes. От. Ну, з того, що я бачила, так, то чомусь для селф-пейнтинга більше Whitefield люди використовують. Конфокальний тільки, якщо хочуть гарні картинки для публікацій, або собі на робочий стіл.
Дуже дякую.
Так, я не бачу якихось ще питань в чаті. І якщо ні в кого вже не залишилось питань, а я думаю, що це логічно, бо в мене теж протягом лекції виникали питання, але вони були покриті буквально на наступному слайді, тому я все прекрасно розумію. Тоді я пропоную завершувати. Запис і транскрипт ми надішлемо поштою. Нам дякують за лекцію, дякую за те, що прийшли. Тож, дякую, Вікторія, що поділилася своїм досвідом. І дякую,
вам дуже дякую за
дякую
запрошення.
всім, хто до нас доєднались. Всім гарного дня!